Ever wonder how the trillions of cells in your body work together to keep you alive and functioning? Cells specialize by developing traits and functions that help organisms survive. This specialization is key for essential processes like movement, breathing, and digestion. In multicellular organisms, cells undergo different stages of specialization. For instance, muscle cells, lung cells, and reproductive cells each serve unique roles in humans. Similarly, plants have specialized cells for different parts like roots, stems, leaves, and flowers, each adapted to perform specific functions. In this video, we'll dive deep into how cell specialization works, the role of stem cells, and why some specialized cells can't reproduce. We'll also explore how stem cells are being used in medical applications. Before we jump in, let's outline our key learning objectives. We will understand cell reproduction and specialization for growth and maintenance, explore the role of stem cells in development and medicine, and learn how cell size influences function and efficiency. Now that we've covered how cells specialize, let's take a closer look at how cell reproduction drives the development of an organism. Cell reproduction is crucial for organism development, starting with the zygote, a single cell formed when sperm and egg combine during fertilization. This zygote contains all the genetic material needed to create a new organism. After fertilization, the zygote begins to divide through mitosis. Initially, the cells are unspecialized, but as they continue to divide, they start specializing into different cell types like muscle, nerve, and blood cells. This process of specialization is known as differentiation. Differentiation occurs when specific genes in a cell's DNA are activated. Even though all cells share the same genetic material, it's the signals they receive that determine which genes are turned on, guiding the cell to take on its role, whether it becomes a skin cell, muscle cell, or something else. Cell signaling plays a key role in this process. Chemical signals, often from morphogens, trigger gene activation. These signals vary across a developing embryo, helping to form distinct structures like the head, limbs, and organs. As development progresses, cells form tissues, organs, and entire systems. However, many specialized cells, such as muscle and nerve cells, lose their ability to divide. In contrast, cells like skin cells can continue to divide throughout life. Together, cell reproduction and differentiation allow a single fertilized egg to transform into a complex organism made up of specialized cells. This fundamental process helps us understand how growth, healing, and bodily function all work. Let's shift our focus to stem cells, which stand out due to their remarkable ability to divide and transform into a variety of different cell types. Unlike most specialized cells that lose the ability to divide, stem cells maintain this potential throughout life. You can find them in specific areas called niches, where they play a role in regenerating hair and skin cells. There's also a wide range in how versatile stem cells can be. Totipotent stem cells, found just after fertilization, are the most flexible. They can form any cell type, including those needed for an entire organism. In contrast, pluripotent stem cells, which appear in early embryos, can become nearly any cell type but are not capable of forming an entire organism. These cells are key players in developing vital tissues like the heart, liver, and brain. As we narrow in on the types of stem cells, multipotent stem cells come next. These are more specialized and give rise to a smaller range of cells. For instance, bone marrow stem cells generate different types of blood cells throughout life. On the other hand, unipotent stem cells are the most specialized. They only produce one cell type, male sperm cells. Stem cells are not just vital for normal development, they have major potential in the medical field as well. Ongoing research claims to harness their power to treat conditions like Parkinson's and diabetes by regenerating damaged tissues. Let's dive into stem cell niches, the specialized areas in the body where stem cells are concentrated. These niches create a supportive environment that allows stem cells to divide and differentiate, which is essential for replenishing cells throughout the body. The function of these niches is to maintain a balance between self-renewal and differentiation, ensuring that a steady supply of stem cells is available for growth and repair. Take bone marrow for example. Here, stem cells generate blood cells, like red and white blood cells. Some of these stem cells stay within the marrow to keep producing new cells, ensuring the body's blood supply remains steady. Hair follicles also provide a good example of stem cell niches in action. In these areas, epithelial stem cells help regenerate hair and skin. These multipotent stem cells are capable of differentiating into various cell types, playing a crucial role in hair growth and skin regeneration. Stem cell niches are also present in the intestines, where they regenerate the lining of the digestive tract, and in the brain, where they help maintain certain brain cells. In each of these places, the niches guide stem cells to specialize into the appropriate cell types for their specific functions. A critical aspect of these niches is the signaling molecules, like growth factors, that guide stem cells in deciding when to divide or differentiate. This signaling ensures that the right balance of stem and specialized cells is maintained.
Now let's explore the different types of stem cells, each with its own unique ability to specialize. These stem cells are categorized based on their potential to differentiate into various cell types: totipotent, pluripotent, multipotent, and unipotent. Totipotent stem cells are the most versatile. They have the ability to form any type of cell in an organism, including those needed to develop the embryo and its supporting structures, like the placenta. These cells appear in the earliest stages right after fertilization. Next, we have pluripotent stem cells. These cells can give rise to almost any type of cell in the body, but they can't form an entire organism. Found in early embryos, pluripotent stem cells can form body tissues, but they're not capable of creating structures like the placenta. As we move toward more specialized types, multipotent stem cells come into play. These cells are more limited in scope and can only develop into specific cell types that are related to their tissue origin. For instance, bone marrow stem cells can produce blood cells but not liver or brain cells. Multipotent cells are essential for maintaining particular tissues. Finally, unipotent stem cells are the most specialized. They are capable of producing only one cell type, such as sperm cells. These cells appear later in development and remain specialized throughout life. Each type of stem cell serves an important function in development and tissue regeneration. While totipotent and pluripotent stem cells are highly flexible, multipotent and unipotent stem cells specialize in maintaining and repairing specific tissues. These stem cells, particularly pluripotent and totipotent, hold great promise in the medical field. Ongoing research is focused on using them to regenerate tissues and replace damaged cells, with embryonic stem cells showing potential in treating diseases like Parkinson's and spinal injuries. However, challenges remain. Ethical concerns surrounding the use of embryonic stem cells and the difficulty in controlling pluripotent stem cells outside the body are major hurdles. Alternatives like induced pluripotent stem cells, which are reprogrammed adult cells, may provide a solution. A deep understanding of stem cells is essential for understanding how organisms grow, develop, and repair themselves. As research advances, stem cells could revolutionize treatments for disease, injuries, and genetic conditions. Now, let's explore how cell size is linked to cell specialization, a crucial factor in how cells carry out their specific functions. The size of a cell is far from random. It is closely tied to the task it performs and the process of specialization that tailors it to be as efficient as possible. Take sperm cells, for instance. These cells are among the smallest in the human body, about 3 micrometers in diameter, but can stretch up to 50 micrometers in length. Their small size is perfectly suited for their role in carrying genetic material and traveling swiftly toward the egg for fertilization. The compact nature of sperm cells allows them to move quickly through the reproductive system to complete their job. In contrast, egg cells are much larger, measuring around 120 micrometers. Their larger size is due to the need to store nutrients that will nourish the embryo after fertilization. While sperm cells are small and streamlined for mobility, egg cells are expansive and resource-rich, supporting the early stages of development. Red blood cells provide another example of how cell size aligns with specialization. These cells measure about 7.5 micrometers in diameter and have a biconcave shape that increases their surface area. This shape is vital for their function of carrying oxygen throughout the body, as a greater surface area allows for more efficient gas exchange, oxygen absorption, and carbon dioxide expulsion. Interestingly, red blood cells also lack a nucleus in organelles like mitochondria. This adaptation provides more space to store hemoglobin, the protein responsible for binding oxygen, making them more efficient in transporting oxygen without consuming it. On the flip side, white blood cells are much larger, around 12 to 15 micrometers in size. These cells defend the body against infections, and their larger size is necessary to accommodate the organelles, like the nucleus, mitochondria, and vesicles, that support processes like phagocytosis, where they engulf and digest harmful microorganisms. Neurons, or nerve cells, also showcase how size correlates with function. Some neurons can be incredibly large, with axons extending up to 1 meter. This length is essential for transmitting electrical impulses over long distances from the brain and spinal cord to muscles and organs throughout the body. Another key factor related to cell size is the surface area to volume ratio, which is critical for a cell's ability to exchange materials with its environment. As cells grow larger, their efficiency at exchanging nutrients, gas, and waste diminishes, since the surface area doesn't grow as quickly as the volume. This is why cells need to maintain an optimal size to ensure they can effectively exchange materials. Specialized cells also show how size can change with their role. For example, skeletal muscle cells can grow very large, up to 12 centimeters in length, which enables them to contract and produce movement. These muscle cells have a large surface area to house the machinery that contracts and releases muscles efficiently. Ultimately, the size, shape, and structure of each cell are intricately tied to its function. Whether it's a small, mobile sperm cell, the large nutrient-filled egg cell, or the oxygen-transporting red blood cell, cell size and specialization work hand in hand to optimize performance. In conclusion, cell size isn't just an incidental trait, it's deeply connected to the cell's specialized function. Each cell's size and structure are tailored to its specific role within the body, ensuring the overall health and proper functioning of the organism. Let's examine the constraints that limit cell size. While cells come in various sizes, there are critical factors that prevent them from growing too large. The primary constraint is the surface area to volume ratio. As a cell increases in size, its volume expands at a faster rate than its surface area. This imbalance restricts the cell's ability to efficiently exchange materials like oxygen and nutrients with its surroundings. To visualize this, think of trying to fill a large box with food. The larger the box, the more difficult it becomes to reach every part of it. Similarly, as the cell grows, some of its internal parts become further away from the surface, making it harder for materials to be exchanged quickly. As the cell's volume increases, it requires more resources and generates more waste. However, if the surface area doesn't expand proportionally, the cell struggles to keep up with the demand for material exchange. This can severely limit its size and efficiency. A smaller cell can more effectively take in nutrients and expel waste, but as it grows larger, it becomes like a factory with too few entry points, less efficient in performing essential functions. That's one reason larger organisms like humans have more cells rather than larger ones. This allows for a better overall surface area to volume ratio and enables more efficient exchanges across the organism. Another factor limiting cell size is the mitotic spindle, which is responsible for helping cells divide. If a cell grows too large, the spindle may struggle to function properly, which can disrupt the division process. When cells get too big, division becomes inefficient and errors can occur, potentially leading to problems like genetic mutations or improper cell functions. Larger cells face challenges in efficiency exchanging gases, nutrients, and waste due to their size. To overcome these limits, they often develop specialized modifications that help maintain an optimal surface area to volume ratio. These modifications can include changes in shape, projections extending outward or inward, strategic positioning near nutrient sources, and how cells are arranged in tissues. Let's look at a couple examples. Red blood cells, or also known as erythrocytes, are a prime example. Their biconcave disc shape increases surface area, allowing for efficient oxygen transport. Their small size and flexibility also enable them to navigate through tiny capillaries, delivering oxygen throughout the body. Another example is a cell in the proximal convoluted tubule of the kidney. These cells are cube-shaped and tightly packed to maximize surface area. They also have microvilli, tiny projections that form a brush border, greatly increasing their surface area for fluid reabsorption. Additionally, many mitochondria supply the energy needed for active transport of substances, ensuring efficient absorption and secretion. These adaptations show how cells can overcome the limitations of size. With the right structural changes, cells can maintain high efficiency, ensuring proper function despite their larger size. This principle of specialization is also evident in lung tissue, where different cells work together to maintain respiratory function. The lung's efficiency relies on the alveolus, its functional unit. These tiny sacs significantly increase the surface area for gas exchange and must endure constant expansion and contraction with each breath. Alveoli are lined by a singular layer of cells and contain three specialized types: type 1 pneumocytes, type 2 pneumocytes, and alveolar macrophages. Type 1 pneumocytes cover 95% of the alveolar surface. Their thin and flat structure maximizes surface area for gas exchange and minimizes diffusion distance. They also share a baseline membrane with capillaries, further reducing the distance gases must travel. Additionally, their tight junctions prevent fluid leakage, ensuring efficient gas exchange. Type 2 pneumocytes, although fewer in number, produce pulmonary surfactant, which reduces surface tension and prevents alveolar collapse. These cells have a cube shape, providing more space for the organelles needed to produce surfactant. Microvilli, facing the alveolar sacs, further increase surface area for secretion. Inside, they contain lamellar bodies that store and release surfactant. Remarkably, type 2 pneumocytes can transform into type 1 cells if necessary, helping maintain the alveolar lining. The alveolar macrophages patrol the surface, keeping the lungs clean by engulfing pathogens and debris crucial for immune defense. Together, these specialized cells allow the lungs to efficiently perform essential tasks: gas exchange, structural support, and immune defense. This collaboration of diverse cell types isn't unique to the lungs. Many tissues in the body rely on specialized cells to carry out their unique roles, showcasing the power of specialization and cooperation for overall function.
Now let's turn our attention to muscle fibers, which are also specialized cells with remarkable adaptations. Starting with striated skeletal muscle cells, these cells are essential for voluntary movement and have some distinctive features. They possess a membrane capable of impulse propagation, meaning they can conduct electrical signals. They're also multinucleated, containing more than one nucleus per cell. This helps meet the high metabolic demands of muscle fibers. In contrast, cardiac muscle fibers, found only in the heart, have their own unique adaptations. While they also have banded striated cells, there are key differences that suit their role in pumping blood throughout the body. These fibers are shorter than skeletal muscle fibers and are composed of branching cells, each with a single nucleus. Importantly, they are connected at their ends by intercalated discs. These intercalated discs allow electrical and mechanical signals to pass between cells efficiently, coordinating the heart's contractions as a unified whole. Instead of dividing to produce new cells, skeletal muscle fibers grow by fusing with supporting satellite cells. When these muscle fibers are severely damaged, they don't undergo the usual process of apoptosis and replacement like other cells. However, despite these unique features, skeletal muscle fibers still share many of the organelles and structural features common to other cells. Let's turn to gametes, the reproductive cells of humans. When a sperm cell fuses with an egg cell, a zygote forms. This zygote undergoes multiple cell divisions, eventually developing into an embryo, then a fetus, and finally an infant. Let's begin with the sperm cell, which has several unique adaptations that support its role in fertilization. Firstly, it's one of the smallest human cells, measuring around 3 micrometers in width and 50 micrometers in length. Its streamlined shape, composed of a head and tail region, is optimized for speed and efficiency. A critical structure in sperm cells is the flagellum, which allows motility. Energy for movement is supplied by mitochondria, located near the flagellum. The head of the sperm contains a specialized structure called the acrosomal vesicle, which releases enzymes to help the sperm penetrate the egg's outer coat during fertilization. Interestingly, sperm cells have very few cytoplasmic organelles, lacking structures like the endoplasmic reticulum, Golgi apparatus, or ribosomes. They are continually produced in vast numbers throughout a male's lifetime and contain a haploid nucleus, carrying half the genetic material. In contrast, the egg cell, or ovum, is one of the largest human cells, measuring about 120 micrometers. Unlike sperm, the egg cell is non-motile and has a spherical shape that is not streamlined. It contains most cytoplasmic organelles and specialized storage structures to support initial embryo development. Now it's time to test your understanding by taking a 10-question quiz. Pause the video if you need extra time to think or write down your answers.